Welcome to the right dose for first one in January 2021. We're going to be talking about the science of COVID-19 vaccines, and we're going to answer some common questions. My name is Ruth Ackerman, and I'm the Director of Professional Development at the Ontario Pharmacists Association. I'm sure you all know me. I've done many of these right doses. This is a pretty exciting one for me. I do the professional development programs for pharmacists and pharmacy technicians at the Ontario Pharmacists Association. I am also a pharmacist by profession. So our agenda tonight is pretty clear. I'm going to have a very brief introduction. Then we're going to do the presentation. I'm going to introduce our speakers, and then they'll take over and do their presentation. And then I'm going to be asking them some questions. Then we'll take Q&A from you, then wrap up the session by 8:25. So my introductory remarks are, first of all, you're all muted. If you're looking for handouts, the handouts for this session were given to you in one of the reminder emails. So if you can go back and get your reminder emails, and you'll be able to see the PDF. There'll be, afterwards, you'll be getting an email from us, and you'll be filling out a survey, a satisfaction survey. At any time, you can go into the Q&A box at the bottom and put your question in, and we will put them all together, and that's where we're going to take questions at the end. So for this webinar, we've had over 1,000 participants, registrants. So we're really excited. We know that this is a top topic around, and we just got some really good news for pharmacists. So just in case you don't know who the Ontario Pharmacists Association is, we advocate on behalf of more than 20,000 pharmacy professionals in Ontario. We're committed to evolving the pharmacy practice and the profession and advocating for excellence in practice and patient care. Our mission is to advance pharmacy professionals as a vital healthcare provider through advocacy, innovation, and support services. And our vision is to be a respected leader in the pharmacy profession focused on health and wellness. So these are some of the stats about who we are and who we look after. Lately, you might have just heard, and that's probably why you're here. We were recently informed by the Ontario government that in preparation for phase 2 of the vaccination distribution plan, pharmacists and regulated pharmacy techs who are trained can inject the COVID-19 vaccine, which is a change to our scope of practice in both professions. The pharmacists are obviously doing injections now, and then we'll be able to expand it to COVID-19, and then we'll have to get regulated pharmacy technicians from the college trained so that they can do the injections, the technical part of the injections. I just want everybody to know that in very shortly, tomorrow, maybe the next day, we will be launching a technician educational program on immunization. It is about 4 CEUs. It trains the pharmacy techs to provide for injections. It's a certified course. It's a CEP accredited course, and it's going to be priced at about $300 for non-members and $200 for members. So look out for that to come out if you're interested in doing it. Before I go to our speakers, I want to go, I want to go to my poll, and I don't know why I can't see my polls. So I'm just going to minimize this, and I'm going to start off with a poll. First poll says, I'm going to ask you, are you planning to participate in injecting patients with COVID-19 vaccine? So here's the poll. So are you going to inject patients with COVID-19? Are you not? Not quite sure, kind of sitting on the fence right now? We have almost 75% of you voting. Okay, I'm going to end it now. And here's the results. We have 57% say yes, 26% say no. Now, could have lots of reasons for that, and not sure yet. And I wonder if at the end of this, we might have those not sure yets going into yeses. Okay. And then I'm going to go to the second one, second poll, which is not showing for me. Oh, okay, maybe we'll do that a little later on. That's odd. I'm just going to try something. No, didn't work. Still poll number one. Okay, so let's go on to our guests for this evening, panelists.
Ruth, poll number 2 is up now.
Oh, it is.
You just have to do the little arrow.
Oh, I see what you're saying. Okay, look at that. Sorry. So the next one is, are you planning on getting COVID-19 vaccine yourself? Yes, no, or not sure. Okay. Oh, you're much faster on the second one. We have 84%, 85%, and it's pretty strong that 81% of you are going to get the vaccine, your shot. So that's really great. That's fascinating. So the next slide is for, just want to introduce our speakers. I have this slide up, but they're going to take over they're going to share their screen. So I'm just going to show you, this is who they are. And then as they're doing that, they're going to share their screen. So the pictures will go away. So I will just talk about briefly who they are. So Dr. Tanya Watts received her PhD in biochemistry at the University of Alberta in 1983, followed by postdoctoral studies in chemistry at Stanford University, California from 1983 to 1986. Dr. Watts joined the University of Toronto as an assistant professor in immunology in 1986. Dr. Watts has had a long-term interest in immunity to viruses, focusing on how T lymphocytes respond to and are regulated during acute and chronic infections, such as influenza virus or HIV. Most recently, she has studied T-cell responses to SARS-CoV-2 in COVID-19 convalescent patients. So Dr. Gomerman received her PhD in immunology at the University of Toronto in 1998. She went on to do a postdoctoral fellowship at Harvard Medical School, then joined Biogen Inc. as a staff scientist in 2000. After 3 years in industry, she returned to academia as an assistant professor immunology at the University of Toronto in 2003. And in 2015 was promoted to full professor. Jen's basic research focuses on autoimmunity, particularly the disease multiple sclerosis. More recently, she has been studying the antibody response to SARS-CoV-2 in patients with COVID-19. And it was very exciting that we have two STEMI professionals. And I just want to point out before I send it off to the panelists that we're wearing our pearls today, our Kamala Harris pearls. So, and hopefully you all know the significance of that, because it was just a social media thing that's happening. So if I want to send it off to you guys to take over the screen, is it working for you?
Yeah, I'm going to just start now. So I hope everyone can see my screen. I can no longer see the chat. So can everyone hear me? I hope I'm loud enough. So I'm great to be with you here this evening. And so I'm going to start off with the first part of the discussion. I'm going to talk about the virus SARS-CoV-2. and how we mount an immune response to the virus, and then how the vaccines are designed to induce a protective response. And then I'll turn over to my colleague, Jen Gommerman, who's going to talk about the clinical trials and some of the evidence of efficacy. So, okay, here's my slide. So as probably most of you know, the human coronavirus, SARS-CoV-2, is the virus that causes the disease COVID-19, which emerged in the late in 2019, first noticed in China as unusual pneumonia. And very quickly, the virus was identified as a coronavirus that was similar in sequence to bat coronaviruses, as well as to the original SARS-1 back in 2002, 2003. Now, SARS is called a coronavirus because of these characteristic crown-like spikes that appear on its surface. And you may have heard in the news of the spike protein. That's the major component of almost all the vaccines, certainly the ones approved in North America. And it's important in attachment of the virus. Encapsulated in this membrane bilayer is the genetic material of the virus, an RNA molecule protected by a nuclear capsid, and it's a single-strand RNA virus. And when it infects cells, it also carries with it its polymerase, which rapidly begins copying its RNA. And most RNA viruses are quite sloppy in copying their RNA. So mutations are actually quite frequent, in contrast to humans, which have DNA, and their DNA polymerase has proofreading function. And so that's why, as long as this virus runs rampant in the community, we're going to see a lot of accumulation of mutations. When a random mutation is deleterious to the virus, that variant just drops out. But if the virus can still produce a full active virus and infect, and if it has any kind of advantage, like better infection, as we're seeing in the UK strain right now, that virus will actually start taking over and displacing the weaker strains. So this is really why we have a very pressing need to get vaccine into people soon, because the longer we allow this disease to run rampant, the more mutations accumulate, and it's really unpredictable what will happen with that. And as you know, to date, we've had more than 90 million infections and more than 2 million deaths in the world. One other thing I wanted to mention is that the virus carries with it genes that help it evade the initial immune response. And that's typical of viruses that's successful in infecting people. They have to be able to take hold initially. But this virus seems to be particularly good at that in some people, and we get quite severe disease in about 20% of people. So how does SARS-CoV-2 spread? It's thought to spread when we cough, talk, or sneeze and release droplets containing the virus, and these small particle droplets lodge in our upper airways, the nose, mouth, or throat. Although they can be spread by direct contact, it's thought that the droplet spread is the most prevalent. Once that virus gets into your upper airways, the virus starts to attach to your airway epithelial cells through this receptor that you've probably heard about, the angiotensin converting enzyme 2, or ACE2. This allows the virus to bind to the cell and be engulfed, where it releases its genetic material, the RNA, and basically takes over the host protein production machinery and translation machinery. So it makes copies of its RNA and makes copies of its proteins and assembles many more copies of the virus that then bud out and then give rise to a localized infection that continues to expand as neighboring cells are infected. However, very quickly after infection, in the very first hours after infection, our immune system starts to sound the alarm. Almost every cell in our body has the ability with intracellular sensors to detect when the viral RNA has entered the cell. And its response is to produce interferons that are secreted from cells, and they produce an antiviral state in the neighboring cells, as well as sounding the alarm to recruit more immune cells into the vicinity. Underlying those epithelial cells are macrophages and other immune cells, and these also have sensors of viral infection and produce the interferons as well as cytokines, again, that call in more cells. So in those first few days after infection, the virus is trying to evade that interferon response, and the immune system is building up. but we really can't eliminate the infection until the adaptive immune system kicks in. So in particular, two kinds of lymphocytes, T cells and B cells, are activated to fight infection and eliminate the virus. So near to the site of infection, those draining lymph nodes, cells that have picked up virus or bits of virus in the site of infection will migrate to the lymph node, and some virus particles can get there too. And these will activate B cells and T cells. So B cells have on their surface antibodies. And if you happen to have a B cell that can stick to that virus, those B cells get activated and become antibody producing cells. And those antibody producing cells go out into the circulation. And with some viral infections or vaccines, these plasma cells reside in the bone marrow for decades, and you can actually have lifelong immunity. Other infections, the immunity is more short-lived, and immunologists are still trying to understand that. But to get a really good antibody response, you actually need help from the T cells that give additional signals to the B cells. And another kind of T cell is very important, the cytotoxic or killer T cell, because that's going to eliminate virus-infected cells. So after these cells are activated in the lymph node, they go back to the site of infection. So when the T cell sees these cells that are actually have become virus factories, they've been infected, the T cell can kill these cells. So although there's some collateral damage and you lose some airway epithelial cells, you get rid of the production of the virus. If you've never seen the virus before on first infection and you don't have antibodies at the ready, you really need those T cells to eliminate the infection. However, 1 to 2 weeks after that infection, you're starting to accumulate antibodies that can basically glom onto the virus and form immune complexes that are readily cleared. In addition, we're really interested in so-called neutralizing antibodies. These neutralizing antibodies bind to the spike protein in such a way that it can no longer attach to the ACE2 receptor. And if you have those antibodies present, you can completely block infection. And so that's generally been the gold standard of immunization and vaccination, is to have a neutralizing antibody response. So how is it that the immune system remembers prior infection? So generally, once you've had a viral infection, you don't get the identical infection a second time. Well, as I mentioned, in the lymph node, we have millions of lymphocytes, T and B cells, and perhaps 1 in a million of those T cells or B cells will have the right specificity for the particular virus. Once it recognizes the virus, it multiplies, it divides many times till you have a large number of lymphocytes, B cells and T cells that combine to give you antibodies and T cells to eliminate the infection. Whoops, I went too fast here. So once those that infection is cleared, so that's shown here, we get rid of the virus once we get the adaptive immune system, so a week or so in most infections. You're still left behind a bigger population of lymphocytes specific for that infection than you had before. If months or even years down the line you get re-exposed to that infection, you've already got a set of cells ready to recognize it. You have more antibodies, more B cells, and more T cells than you had the first time you saw it, and you get a much more rapid response. And that actually prevents infection generally, so you would, if anything, have no symptoms at all and perhaps little or no viral replication, and you're protected. As we'll hear, this is the same principle that a vaccine works on. I should also mention the secondary response is bigger and also tends to not go down as low and to be more sustained. And that's going to be the principle of boosting in a vaccine as well. So let's switch now to the vaccine. How do vaccines work? Well, the vaccines against viruses work when you give a person an inactivated or attenuated virus, or just part of the virus that's recognized by the immune system in such a way that you can mount an immune response and give yourself those T cells, B cells, and antibodies so that when you encounter the actual virus, you're protected. And so it's tricking the immune system into thinking there's infection when there's not. And I'm sure I don't need to tell this audience that vaccination is one of the best medical interventions that have been devised by man, leading to the eradication of smallpox and as well very good vaccines against deadly childhood diseases such as polio and measles and other infections. So let's focus in on the current vaccines for SARS-CoV-2. We'll start with the RNA vaccines because we have 2 vaccines based on RNA that are currently approved in Canada. So these are given by intramuscular injection, and they consist of a lipid nanoparticle encapsulating a single strand of RNA. And rather than encode an entire virus, they only encode the spike protein. And no virus is used in the production of this RNA, but the lipid nanoparticle protects the RNA, which is quite a fragile molecule, and also facilitates uptake by phagocytic cells at the site of infection. Once inside the cell, the RNA gets translated into spike proteins, and this is a membrane protein, so it will end up getting to the cell surface of these cells and being displayed on the surface to the immune system. It's worth noting that RNA is only transiently expressed in the cell. It can't replicate. It's an unstable molecule that gets rapidly degraded. And so there's only a transient production of this spike. But it's sufficient that some of these cells are going to go to the nearest lymph node. And just like if we'd had a viral infection, we will get activation of B cells and antibody production, as well as T cells. And now these cells will go back into the circulation. And if subsequently you become exposed to the virus, the actual virus, you've already got a population of T cells that can kill any infected cells. And if you have neutralizing antibodies, you can actually completely prevent the secondary infection. And really the T cells at this point become a backup if any viruses escape the neutralization by the antibodies. And I mentioned earlier about boosting. So the RNA vaccines from Moderna and Pfizer require 2 injections. The first injection gives you a pretty good immune response that's about 84% protection after about 11 days. And then at day 21 to 28, a second injection is given. And this actually enhances the level of antibody and T cell response. And it's thought that this is going to give rise to longer lasting immunity, although we don't have long-term data to know how long that will be. And so the result of these 2 injections is that people will have more T cells, B cells, and antibodies to protect against infection. So I mentioned the RNA vaccine. So there's 2 approved in Canada from Moderna and Pfizer. Another approach to vaccination for SARS-CoV-2 is to use a recombinant adenovirus. These are common cold viruses, and they've been gutted of their 2 essential genes so they can't replicate in cells. And those genes have been replaced with the spike protein gene. And so when these viruses can get into cells once, but they can't reinfect, they basically release the DNA, and much the same as the RNA, they'll give rise to an RNA transcript and spike protein. The first one approved is actually using a chimp adenovirus. And that was chosen deliberately because humans wouldn't have been exposed to chimp adenovirus, so that we wouldn't have antibodies that could neutralize the vaccine. The other one that's coming up a little bit later, so the chimp one, Chadox, is coming from AstraZeneca in collaboration with Oxford University. And shortly behind that one is one from Johnson & Johnson that was in collaboration with Harvard and the NIH. And that uses an adenovirus 26, and only about 10% of our population has been exposed to that virus. Also in phase 3 trials, are some vaccines based on just purifying the S protein and emulsifying it in an adjuvant, which is an immune-enhancing substance that helps promote uptake and immune stimulation. Phase 1/2 trials of this protein-based vaccine look very, very strong, and we're waiting for phase 3. You may also have heard in the news about the Medicago vaccine, which is a made in Canada vaccine. They're going to be starting phase 3 trials, I think, within the next few weeks. And this is interesting because they use tobacco plants. And last year, the company made 10 million doses of flu vaccine this way. So it's a really good method for large-scale production. And basically, you give the gene for the S spike to these tobacco plants, and they cleverly produce a lipid vesicle with the spike protein on the surface referred to as a virus-like particle, which is very good at tricking the immune system into thinking there's an infection when it's not. So all of these vaccines have in common that they don't use any actual live virus for coronavirus for the production. The RNA virus is actually chemically synthesis, so there's no cells involved. And these other proteins, of course, need some sort of cell line to produce the product. So I think I'm going to stop there. I'm going to stop sharing, and then Dr. Gommerman is going to go through how these vaccines were approved.
There we go. Sorry, I was muted. All right, I'll start over. So to give you a sense of the trajectory of what happens when you have a new vaccine that has to be tested in order to eventually be approved and used in the public, I'm going to walk you through the different phases of clinical trials. So in very brief, the first steps are obviously in the wet lab itself, where the scientists are actually designing the best way to deliver the vaccine. In the case of the mRNA vaccines, that was a very rapid step because basically as soon as they had the sequence for SARS-CoV-2, they were able to insert it into their mRNA vectors in order to make the mRNA vaccines. So they do these steps in vitro to determine whether the vaccines will be effective. And then they also use animal models, for example, mice that have been engineered to be susceptible to SARS-CoV-2 infection, or non-human primate models. And those models can be used to also challenge the animal following vaccination with a dose of SARS-CoV-2 to determine if the animal can clear the virus and whether they get COVID-19. And if the vaccine candidates look good in the preclinical studies, then they start to test them in humans. And in the first phase, really all they're doing is checking for safety. And what they do is they take the vaccine and they start at a low dose, and then they go a little bit higher in other subjects. It's called a dose escalating study in order to determine the safe dose for the vaccine and what dose gives the best efficacy. And in these cases, they're looking at things like antibody production or T-cell immunity to determine the efficacy. And then they open up the trial to more people. Here you're looking at hundreds of people in order to ascertain the safety in a broader population. So you want to be able to look at safety across age groups, for example, and that you can do in phase 2. And again, you can get information on the immune response in phase 2, whether the vaccine is eliciting a good antibody response or a good T-cell response. And then lastly, provided the safety data look good, you then expand into phase 3 trials, which involve thousands of people from broad ranging populations, different ages, different ethnicities. In the case of the 2 mRNA vaccines, these involve tens of thousands of people. So for example, one of the trials put 15,000 people in the placebo group and 15,000 people in the vaccine group. And I should mention that this is what's called a randomized double-blind trial, so an RCT, randomized control trial, and the people who are the company people don't know who's who. That's blinded to them, and then they run the trial, and the trial has a very clear endpoint, and it is, for example, when 100 people get COVID-19, they would stop the trial and then unblind the trial and determine whether those 100 people had received the placebo or whether they had received the vaccine. And that's what would determine the efficacy. And that's how those particular trials were designed. And if the data for efficacy look good and the safety profile is acceptable, then what health regulatory bodies such as Health Canada or the FDA do is then they look at that data, and if it meets what they feel are the criteria needed to vaccinate, so in Canada, this would be Health Canada, obviously, then they would give regulatory approval for that vaccine. And at that point, the vaccine can be used among the public. But it doesn't stop there. People continue to be monitored, not only the people who were in the trials, but as new people get the vaccine, and now we're looking at over 100,000 people, hundreds of thousands of people have been vaccinated, we're continuing to monitor the people who are getting the vaccine, because sometimes adverse events can be extremely rare. And so you really want to know whether there's any adverse events associated with delivery of the vaccine. I should also mention that it might have felt like this trial process was really fast, but all of these steps are the same, irrespective of what kind of trial you're doing, whether it's a trial for a drug or a trial for a different vaccine. One of the reasons it was quick was because they didn't have to rationalize doing this. They didn't have to spend money on marketing or anything like that. Obviously, this was a really big priority for the world. And then when the phase 3 trials ran, it happened to be in the fall. Many of the trial subjects were in the US, in hotspots. And so getting that 100 people to develop COVID-19 actually, unfortunately, didn't take very long. And so that was sort of a silver lining, because then they could have a look at the data as soon as they reached that target number of people with COVID-19. So all of these steps were respected and were done just as normal with these vaccines that are currently being tested and the ones that have gone through up to phase 3 and beyond. So Canada has secured a number of vaccine candidates, including the mRNA vaccines, which I'll show you the results from those today, because those are the only two that have been been at this point approved. Coming down the pipe are these other candidates that Dr. Watts mentioned, and I'll show some data on the J&J vaccine, which looks really promising, but many of these are really looking good. So I think we're gonna have a really nice, robust, healthy pipeline of vaccines to get Canadians vaccinated and achieve herd immunity. So coming back to this RNA vaccine, because these are the two that are, excuse me, are approved. Let's talk about the data for these two RNA vaccines. So as Dr. Watts mentioned, RNA vaccines contain mRNA, and this encodes the recipe to make the SARS-CoV-2 spike protein. And it's inside this lipid particle, which is really easy for uptake into cells. And then the particle is degraded, the mRNA is made into protein, translated into protein and expressed. And the mRNA itself does not last very long because it's unstable. And the two vaccines that are mRNA-based are the Pfizer BioNTech vaccine and the Moderna vaccine that you've heard so much about in the news. I should also mention that none of the material from those mRNA vaccines actually enters the nucleus. So this is all happening in the cytosol of the cells that take up the mRNA. So as I mentioned, large phase 3 clinical trials were enrolled tens of thousands of people by both Moderna and Pfizer BioNTech. And the primary endpoint, this is sort of trial language, the primary endpoint of the trial was to monitor the development of COVID-19 illness. And if we were to imagine what a perfect trial would look like, it would look like this. The placebo group over time would show evidence of these people acquiring COVID-19 illness, and the vaccine group would hopefully not have COVID-19 illness over time. So this would be the theoretical perfect world data that we'd love to see in a trial. And I think it was a really amazing moment for scientists when this is what the data looked like. So this is the real data from the Pfizer trial, and the Moderna data are very similar. And you've heard the number 95%. And that means that when at the end of the trial, when they looked at those subjects who experienced COVID-19, 95% of them were in the placebo arm of the trial and not the vaccine arm of the trial. So the vaccine arm is down here. So this was a wonderful, amazing feat for science. And if you actually back up and look in the first 3 weeks after the first shot of the vaccines, this is before the boost, already you're starting to see a separation of those lines. And so that means that even after one shot, there's efficacy as a consequence of vaccination. And this trial was, as I mentioned, enrolled a very diverse set of participants. And the other good observation from this trial is if we just look at severe COVID-19, the form of COVID-19 that ends up in the hospital and really people who are extremely ill, no severe COVID-19 was seen in the vaccine arms of these two trials. So a lot of people have asked me why, if the vaccine is just a part of the virus, would your immune response not be as good? And the answer so far is that no, your immune response is just as good with this vaccine as what you would get with the natural infection. And so this is some of the phase 1 study data where antibody levels to antibodies to the virus, in other words, neutralizing antibodies that Dr. Watts mentioned, were measured. And they used this gold standard assay where they looked at the antibodies that were generated in the people who were vaccinated after 43 days. And those antibodies, if they can block viral entry into host cells, they can tabulate how much of that antibody is needed, and it's called a titer. And as you can see, it didn't matter what dose the participants got, but a lot of these neutralizing antibodies were generated. These are extremely good titers of neutralizing antibodies. And this is the amount of neutralizing antibody that's made by someone who's had the natural infection and has recovered, in other words, convalescent patients. So if anything, the level of antibody, well, we can at least say it was equivalent in the vaccinated group, and it certainly wasn't less. If anything, it looks even a bit higher. So that was really good news. The second thing that I often get asked is, well, how long is that immunity going to last? Are we going to have to just keep revaccinating all the time? And the short answer is we don't have that data yet because it hasn't been that long since the vaccines have been rolled out. But what's been ongoing is that as patients got enrolled in these trials, they keep sampling them through time. And now we have data out to 119 days, so 4 months post-vaccine and boost. And when you look at subjects, and it didn't seem to matter what age they were, all of them are keeping those antibodies in their blood. So again, these are antibodies that are capable of neutralizing the virus. And you can see that they're generally pretty stable through that 4-month period. So that's good news too. That means the immune response is going to likely last a good long time after the vaccination. And that's just the antibody response. Remember too, that the vaccine is also going to prime cells, T cells, and also going to make memory B cells and memory T cells. And so this is only one of the pieces of our artillery against the virus. There's other things the immune system can do to deal with the virus. So once you get vaccinated, people ask, well, can I just throw my mask away and go back to life as normal? I just want to remind people that getting COVID-19 is getting the illness as a consequence of being infected with SARS-CoV-2. We know that lots of people who get infected are asymptomatic and they never really develop symptoms. So they don't get COVID-19, but they've still been infected. Now the trials were designed I think quite rationally to really look at the rates of COVID-19 as a consequence of vaccination. So they had some hints of who was going to be infected at some of the visits that the subjects went through, but in general, they couldn't really take into account how many people were getting infected with SARS-CoV-2 on the trial. So that answer is still unknown. However, there is some hints that the vaccines may provide protection against infection and would therefore block transmission. That's what we call sterilizing immunity. And the reason we know that is that if you look 4 weeks after the first shot, it seemed that there were a higher number of people that they happened to catch at that moment through the nasopharyngeal swabs that were positive for SARS-CoV-2 than those in the vaccine group. But it's a small number because it's just those people that happen to be testing at that moment. So the short answer is when we are vaccinated, we should not assume that we can't transmit, although I think it's a good chance that transmission will be reduced. And besides, no one is going to be wearing a sign saying, I just got vaccinated, and not everyone's getting vaccinated at once. So I think it's also, I think it's a good thing to wear the mask and continue social distancing and other public health measures. And hopefully, once enough people are vaccinated, we're going to achieve what's called herd immunity. So what is this? This is a state where there's enough immunity in a population that the virus can't hop around from person to person anymore, because that's really the goal of the virus. The goal of the virus is not to kill us. The goal of the virus is to move from person to person. And this one does it pretty well. So what we were at in the spring was this. We had very few people who had ever been infected with this virus, only a few, and most of them in China, because the virus had not come here to Canada yet until around March, and then it started to spread. And so at this point in the pandemic, there was very little herd immunity. This is a novel virus, so there's really no immunity within our population. We're extremely vulnerable. But then as the pandemic progressed, more and more people got infected and developed immunity, but not enough to actually generate herd immunity. Like all these people in red here are still susceptible. And to really achieve herd immunity, where there's enough people who have immunity to the virus, if we relied on natural infections, that would take a very long time and there would be a lot of collateral damage. However, the vaccine accelerates that calculus, and we can achieve herd immunity through vaccination much more quickly and much more safely. And sorry, I should mention the estimate at the moment is that we need about 70% herd immunity to get there, but that's really an estimate. It might end up being higher. And so really we need to encourage people to take these amazing vaccines so that we can get to herd immunity, effective herd immunity as quickly as possible. There are some side effects with vaccination. Vaccinations are meant to stimulate the immune system, and so that's something that we feel when we get the injection. This vaccine is considered in the category as reactogenic, which is the same category as, for example, the Shingrix vaccine. So you will get some side effects due to immune activations. You'll get a sore arm, that's the most common, but you might have some fatigue or a headache. And that's all very treatable with acetaminophen or NSAIDs. In terms of more serious side effects and adverse events, there was a report that there was a little bit of more Bell's palsy in the vaccine group compared to placebo, but it was really small numbers. And in fact, the numbers even in the vaccine group were no greater than what you would see in the general population. So we don't think that Bell's palsy at this moment in time is a risk that we need to worry about. And then Guillain-Barré syndrome is something that has been observed with vaccines in the past, although never quite firmly associated with vaccines. It has not appeared in any person who's received a COVID-19 vaccine or transverse myelitis. Allergic reactions have been reported, but they're still very rare. They're about 1 in 100,000 people. Basic recommendation is that pharmacists would observe a subject for 15 minutes after their vaccination and 30 minutes if the subject has a history of severe allergic reactions. So what's coming up? This is the J&J vaccine. This is phase 1/2 data. And what I was really excited about regarding these data was that if you compare people who got 1 dose of vaccine, so they got a low dose and no second dose, versus those who got 2 doses of the vaccine, the level of antibody is very similar between these 2 groups, which means, and that's all the way out to day 71, which means that for this vaccine, 1 dose might be enough. And that would be great because that would make it easier on logistically to administer the vaccine, knowing that you don't have to book a return visit. So that's exciting, and we're looking forward to the phase 3 data on the J&J, which are ongoing. And really, I think one of the most amazing things for me is that a year ago, we didn't have any vaccines to SARS-CoV-2, and look at this vaccine pipeline that we have now. So this is really an amazing international scientific collaboration, and this is a very robust pipeline, so that we'll be in a position to offer, hopefully sooner than later, vaccines to all Canadians. And you can find this on the vaccine tracker for New York Times. So these are some of the literature that I've cited as I went along. You're welcome to check it out in the handout. And before we get to questions, I just want to, on behalf of Dr. Watson, I thank Ruth and the Ontario Pharmacy Association for the opportunity to speak to you tonight. I think this is probably one of the most important talks I'm giving this year, because you guys are going to be on the front lines delivering the vaccine, and we're counting on you to be part of the fight. And so thank you very much for listening.
Oh, that was awesome. That was so I kind of remember that. I think I learned that in school. Don't ask, I hope there's no test at the end. So it was very good, very scientific, exactly what we were looking for when talking to people. So we got a ton of questions, and
I've been trying to answer some in the Q&A, but I thought we could do some verbally too. So
absolutely. So one of the things I thought, there was a couple of them that I would like to ask, sort of to start off with. Some of the things that you didn't touch on. There's certain classes of people who have certain comorbidities, perhaps you could call it that. So pregnancy, if you could comment on whether COVID vaccine should be given to a person who's pregnant.
Do you want to take that one, Jen?
Sure.
Yeah.
So Dr. Watson and I had a look through the recommendations from the vaccine panel. Actually, was it from PHAC? I can't remember, Tanya.
It might have been NACI, National Advisory Committee on Immunization. Yeah.
Yes, thank you. And so unfortunately, pregnant women were not included in the trials. So we don't have that information, but we know that pregnant women do carry some risk from COVID-19. And so we don't want to see pregnant women getting COVID-19. So basically you should be having that conversation with your OBGYN and your primary care physician to determine for you specifically if this is an appropriate time to get vaccinated. So that's unfortunately all we can say right now, because the trials did not include that particular subject group.
Mm-hmm. Oh, thank you. We often see patients in our pharmacies who are immunosuppressed through medications, cancer therapy, etc. Maybe you could comment on that. Should they get it?
Yeah, so that's really an important point, because unfortunately the clinical trials are all with healthy people, so we don't have data. We're actually starting a study, an observational study with Mount Sinai, Toronto Western, and Women's College on this issue for people with autoimmune and conditions and inflammatory bowel disease who take immunomodulators, because we really want to know. But my view, if you look at the effects of the actual infection, it's very inflammatory. And even if we look at the history of flu's worse actually for autoimmune disease if you get the actual infection. So people always say, oh, a vaccine is going to give me inflammation. Maybe it's not good for autoimmunity, but you'll get much more inflammation from the virus. So my recommendation is, yes, you take the vaccine if you have an autoimmune condition. If you're on immune modulation, we don't know how good your immune response is, but even if you get a partial effect, that's got to be better than nothing. If you get exposed to the virus, it could lessen your disease. So we really want to know how good those people's immune response is. But obviously, I think they may want to consult their physician. But my opinion would be to take the vaccine. They're more vulnerable. And I guess another question we may be asked is about, dose 1 versus dose 2 and delaying it. I think for that population, dose 2 is really important, because if you have a weaker response, you're going to need that boosting even more. So,
So maybe you could keep going with that space, because that's the challenge that a lot of the governments have today with the not enough vaccines. So they're stretching out that recommended studied interval longer and longer. So maybe comment on that.
Right. So we've had quite a discussion on this on the Ontario COVID-19 Science Table, and they've actually put out a document on that. It's available on their webpage. So first of all, boosting, or the second response, is important for 2 reasons: increase the response and increase the duration. Now, we do know that you can wait quite a long time and boost. So measles vaccine, you get it when you're young, you get it again school age. So you'll still get the boost. So it's not that the boosting won't work. The real question is, how much are you protected after 1 dose? We know after about 11 days after the first dose, people are about 84% protected. We know that that protection lasts for, well, we know it's the immune system, the immune response stays high till day 21 when you get the next dose. We don't really know how quickly it'll fall off if you don't get the second dose, but there's some evidence from not identical vaccine, but a similar one that it's pretty good till 42 days. So that's why for Ontario, the recommendation was there's reasonable evidence that if most people waited 42 days, they'll still get the effect of boosting. They won't be at that 95% protection, but they'll be at, 75 to 85%. And if you actually weigh the lives saved by more people getting dose 1 versus moving from 84 to 95% over a few weeks, I think the cost benefit analysis was that you try to spread the vaccine out with the anticipation that the supply will catch up and they will get the vaccine at about 6 weeks. Quebec saying up to 3 months. I'm sure they'll get a boosting effect at 3 months. It's really just a trade-off between how many people might start to lose immunity or not be fully protected, how many people really needed that boost, because we're all individuals. I think the clinical trial tells us a set of people, fairly healthy, average response. When you go out to millions, you're going to have more variability. So I think it's just a benefit analysis of trying to save the most lives. I think everybody should get 2 doses eventually, but giving out that dose 1 rather than sitting on it for 3 weeks, and then hoping that by 6 weeks, and we know we're having a little delay now, but the anticipation is that's a short term and we'll start to get more vaccine coming in a few weeks.
And I'd like to just jump in and add a couple things. So the day 21 and day 28 design for the 2 trials, it was designed to get an answer, because if they said we're going to do a prime and then we're going to do boost 3 months later, which would probably have been more typical, well, then we would have been waiting that extra time for the answer to the trial, and that would have also delayed decisions on whether to approve these. So, the prime boost schedule may have been affected by that calculus. And also, I think it's like Dr. Watts said, not every solution will fit everyone. I think if you have a really weak immune system, if you're elderly or you're immunocompromised, I think those people should get the manufacturer's schedule for the vaccine. But for healthy people, there's emerging data shows that the efficacy is pretty good after 1 dose. If you did become infected with COVID-19 after 1 dose, that's kind of like your boost. So you might get infected, and you might even get a little bit sick, but you're likely not going to get seriously ill. That's the thinking, because you've already had an exposure, so you're going to make a memory response. So it's like Dr. Watts said, it's a bit of a tactical decision. And if we didn't have a shortage of vaccines and we weren't in a surge and there weren't variants, we wouldn't be having this conversation. We would just do exactly like the manufacturer said. But these are extraordinary circumstances.
Absolutely. The pharmacists are going to start giving COVID-19 vaccines to their patients, and some of their patients might have a current active either viral or bacterial infection. So should it be given? Should we wait? What does that do to the immune response?
Well, I think, normally, I'm not a medical doctor, so I'm an immunologist. So I think people often don't recommend having a new vaccine when you're sick, because you're looking for side effects and things. So I think it's really, an individual choice, how desperate you are for your vaccine and things like that. But probably consulting their physician is probably the best bet.
I also don't think we want to see sick people in line for vaccines.
Well, right.
Oh, yes, you wouldn't want to see someone with COVID-19. Exactly.
So people, that's one thing to really point out, that when people have these vaccines, they're not instantly protected. It's going to take them about 11 days. So, from the Israelis, which are really far ahead, they've had a bunch of cases after vaccination, and they're saying, oh, well, the vaccine isn't working. But in fact, if those people were already exposed or get exposed in the next week, they're going to get sick, even though the vaccine's perfectly good. It just hasn't had time. So I think we really need to make sure we counsel people to continue to wear masks and hand wash and physically distance. And also we could talk, maybe Jen wants to talk about that, the issue of, we don't actually have complete data yet on that we know the vaccine protects the individual against clinical disease. We have some data that it protects against asymptomatic infection, but through very small numbers. We don't completely know yet that it stops you from spreading the virus, and we're waiting for that epidemiological data. So I don't know if you want to add to that, Jen.
No, I think we'll find out from the epidemiology.
Yeah.
Okay, co-administration with other vaccines.
Hmm.
I want to get a flu shot. It might be October, right? When I get a flu shot in here and a COVID shot here.
Yeah, I don't know if NASA's actually made a decision on that. I know that this is a topic of discussion. Children come and get several vaccines at once, right? It's not unusual to have more than one vaccine. From an immunological point of view, our immune system has a lot of capacity to recognize many different antigens. So from that point of view, it's not theoretically bad, but I think most people prefer to separate the vaccines just in case they get a side effect to one. If you had a reaction, do you know if you should get your second shot or not? Things like that.
Yeah, I think there are a number, I'll just let everybody, the audience know that there are some resources that actually do talk about that.
Yeah.
One of the examples would be Shingrix vaccine, the vaccine for shingles. There is meant to be a 2-week window between the two vaccines. So,
Okay.
so there, I think if people, I have a list of references, you have a list of references where people can go get more information about that.
Just a word about the Shingrix vaccine that I meant to bring this up as an example. I was looking up the schedule for Shingrix, and the boost can happen anywhere from 2 to 6 months.
Yeah, exactly. Yeah.
And so you can see how there's a big range, but Shingrix wasn't developed in the midst of a shingles pandemic. So, that I think that's a good example of, there's probably a fair bit of flexibility in that boost, maybe not for people with weak immune systems.
Yeah, I think that's what most people expect. And the Shingrix vaccine's quite reactogenic too. People get particularly sore arms, fever. So again, I think that's the recommendation. You don't want to be having, confounding these side effects. So.
Absolutely. Are there any absolute contraindications to getting the vaccine? Are there people that should not get that vaccine?
I think the one interaction is if you have an anaphylactic reaction to the first dose, you don't get the second dose. That's certainly being written in the NACI guidelines, but there was suggestion if people are prone to anaphylaxis after vaccines that they not take it. So.
Okay. Oh yes. If a person has been affected by COVID-19, so they were and recovered, should they get the vaccine, and would they have a different response?
So we don't have data yet on that, but they should be immune, so the vaccine should boost their immunity. I don't think they should be in a big hurry ahead of the unexposed, because with the data says they should be protected for about 8 months so far. We have data. Sorry, Jen, you were going to say something.
Yeah, well, I would just say that exactly that. If you have had confirmation that you had SARS-CoV-2 infection, then you can probably go to the end of the line. But a lot of people came to me in the spring saying, please measure my antibodies because I was sick with something and I'm sure I had COVID. And then they were negative. And I think that's just human psychology. When you're talking about COVID all the time in the media and you had some illness, you may not have had SARS-CoV-2. So I think we should counsel people, yes, to get the vaccination. And if they did have that positive PCR test and they knew for sure they were infected, then they can deprioritize themselves with respect to the queue.
This one's kind of a good one. So I get the COVID-19 vaccine and then I get COVID, either because I was already infected or later on. Can I transmit to others?
Yeah. I think if you get the infection, you can transmit. Yeah.
Yeah, because you've got the infection, and then the virus we don't know about transmission, whether.
Yeah, I mean, as long as it's replicating in your upper airways, you're going to be coughing out virus. So,
yeah, that's the other thing. transmission, you talked about aerosol droplets. So well, I think they're called fomites. So everyone's washing their groceries, or were washing their groceries. I don't know how they're doing now. Maybe comment a little bit on that, or if you know, if you've done studies or.
I mean, there's been a lot of debate in the literature, is it, you know, aerosols or droplets, but I think they're really a continuum, you know, little droplets stay in the air longer and go further, big droplets fall out faster. And, you know, I don't think there's not some cutoff exactly what do you call a small droplet versus an aerosol. So I think the, you know, something like TB, which is an aerosol, it takes very little to transmit. We're seeing these new variants are transmitting very easily. So I think that's sort of debatable whether you call it aerosol or droplet. But generally, I thought the consensus was it's a bit more like flu with droplet spread. I think the consensus on, you know, contact, like somebody sneeze on a surface, you touch the doorknob right after them, touch your face, you could catch it that way. There have been a lot of studies trying to measure by PCR that they get virus off surfaces. But the question is, what is the infected dose? So I think it's theoretically possible to get the virus from a surface, but it's not, I don't think it's the main route. It's clearly the main route is community spread and, you know, household contacts, breathing the same air. I personally don't wipe down all my groceries, but I wash my hands after putting them away and wipe the fridge handles now and again. So.
Well, better safe than sorry. It's probably a good thing to do. You touched briefly on the mutations that we're seeing. Can you explain what that means when they say it's more transmissible? And can you explain, how do the vaccines that we have, are they going to still be effective against the mutant?
So the so-called UK variant, which we're calling B117, I think it's more appropriate to call it that, has a mutation in it that actually changes the spike, the contact area between that spike protein and the ACE2, that is the host receptor that the virus uses to get inside of host cells. So the hypothesis right now is that that change has made it easier for the virus to enter cells. And I think that's a reasonable hypothesis because that would imply that the virus has mutated to do exactly what it wants to do, which is to move from host to host. That's its goal. So that's one of the variants, and there's another from South Africa that's being investigated. There's another one in California I just read about that's being investigated. I think what took us a little by surprise is that coronavirus itself is not a virus that mutates a lot, not as much as flu, for example. And so I think, I don't know about Dr. Watts, but I was taken a little by surprise when all of a sudden all these variants were coming up. But then when you think about it, it makes sense because we've had about 100 million people infected. And so it was really only a matter of time that these variants would emerge because of that problem with herd immunity. We didn't have any herd immunity at the beginning of the pandemic. So it can just really rip right through our population, and then it becomes a numbers game and something is going to emerge. And actually that should make us extremely motivated to get everyone vaccinated. Exactly, because the longer this goes around and runs rampant, the more opportunity there will be for it to mutate, potentially an immunocompromised person. We're not too sure how it happens, but we want to stop this cycle as soon as possible.
I would
now, in terms of whether it evades the vaccine, so far there's no evidence for that. The reports I'm seeing that are granted not yet peer-reviewed have shown that if you take antibodies from people serum from people who were vaccinated, or people who had the original variant, the Wuhan variant, that serum can still stop the virus from entering into human cells. So it doesn't appear, and that's only the antibody response. We still have the whole cellular immune response that's capable of coming to the rescue as well. So far there doesn't appear to be significant evasion of the immune response. And so that would tell us that the vaccine that we have right now should be good against the variants that are circulating at this moment. But again, we have to hurry because we don't want to leave that one to chance either. So we really got to make sure we get this under control.
Yeah, I think scientists think, so influenza, as you know, evades the immune response by mutating very quickly. And sometimes the vaccine that we make in the spring, by the following spring, is no good. And that's because the way influenza attaches to cells is through a little small molecule of sialic acid that goes into a cleft. So the antibodies can't bind there, they bind around it. So the influenza can mutate the residues around and still have its binding site. Whereas the coronavirus uses this tip of the spike to bind this protein, the ACE protein. And so there's actually a lot of contact that the antibodies can have and still block the virus. So it's going to be harder for that virus to evade the immune response. And also the size of the S spike in the vaccine. I think it's about 1,500 amino acids. So, you know, if it's accumulated 25 amino acid changes, it's going to take a lot to get rid of all the immune response. So.
And remember too, that the immune response isn't just one little spot that it's going to target on the spike. It's going to make what we call a polyclonal immune response, which means it's going to make antibodies that can see all parts of the spike. And that's on purpose in case the virus does change. And the same is true for the T cells. So our immune system is designed to deal with this. But let's not push the envelope. Let's get people vaccinated.
Yeah. I think the biggest issue is the variants that are spreading faster. They don't seem to be more virulent, but no. If they spread faster, they kill more people because they're just getting that many more infected. The rate of in the UK right now is really scary. So I think that's the big reason to get the vaccine out quickly. Yeah. Mm-hmm. If we can, we hope.
Some questions about children. So should we not, because they haven't been directly studied. Do you know what people are thinking now about getting children?
I think there's an ongoing trial for 16 to 18-year-olds right now. So that data should come out soon. But I guess we're just gonna have to wait for those data.
I think it is going to wait. I mean, the death rates are lower in children, but they are still getting this multi-inflammatory syndrome in rare cases. And so I think eventually they'll do trials at younger age groups, but at this point, they're not recommending the vaccine for children. So.
Lots of immunocompromised. So after receiving the vaccine, should people still do surveillance testing for COVID-19 when visiting their family in long-term care? Do they need to continue to have surveillance testing until herd immunity, or stop surveillance testing 14 days after receiving?
That's a good question. Certainly initially, I think they need surveillance. I think it'll be a policy that will have to be put in place by long-term care, but no vaccine is 100%. So these trials say 95% of individuals after 2 doses in an ideal, healthy population. So, I think if you're going into a vulnerable setting and they are able to do testing, they probably will want to maintain it because, that means 5 out of 100 might not have been protected.
Mm-hmm. Somebody mentioned, and I'm not sure whether you know about this or not, allergy to propylene glycol as a contraindication.
I think it was polyethylene glycol, was it? Or?
Well, I think it's polyethylene glycol that's in it.
Yeah.
And if somebody has an allergic allergy to propylene glycol, then should they take it?
I don't know. Yeah.
Yeah, okay, that's fine. Let's see. I don't know whether you have this expertise, but there's a lot of conspiracy theories going around that it affects your fertility, getting the vaccine, and all kinds of. We don't know enough because there may be some long-term side effects. I always get that.
Well, we don't know of a single example. I think vaccine problems happen in the first 2 weeks. I don't know of a single example of late effects of a vaccine. And so when you think about a virus, it's replicating, it's going everywhere in your body. And here with the vaccine, you've got this local administration into the muscle. It's short-lived. It's just not, I think there's no plausible reason for any of these fears. So It's true there weren't very many pregnant women in the trials, although accidentally there were about.
Yeah, actually, I was there
Yeah.
were 10 in each group.
Yeah.
Just by chance,
Yeah.
and so not enough to really make any conclusions.
Mm-hmm.
And here's another person said, I've heard a number of people in the medical community mention that they are waiting for the AstraZeneca vaccine, as those utilize the viral vector method with the adenovirus, which is what many vaccines in the past are made of, versus mRNA, which is a new technology that has never been used on vaccines before.
Well, actually, the adenovirus, I think the only licensed one is the Ebola virus. So I wouldn't say it's been widely used certainly not in North America. I personally will take the RNA vaccine if it becomes available to me. They look really good. And because it's so simple, there's nothing to worry about it. It's a really very simple vaccine. The adenovirus, I mean, they're quite effective in delivering the DNA to the cell. And so they should be effective. I think if anything, the data is better on the Moderna and Pfizer than the AstraZeneca, but the Johnson & Johnson looks even better. So, but they're all very good. I mean, I think if you were to say to me, you can have this one or that one, it's available, I would take the first available personally.
Me too. And it could also be that people are looking forward to the like, for example, the J&J vaccine, the storage requirements are much more straightforward. I think it can be kept in the fridge. And the J&J one looks like it's working with one dose. So there are reasons to look forward to these vaccines too, but not because they're necessarily safer or better.
And I think, eventually it could be very well, people have asked me that, maybe you get an adenovirus vaccine first, and if we do turn out to need boosting in 3 or 4 years, if it sticks around, maybe you get an RNA vaccine later. The vaccine companies don't test that. They test their own in the prime boost. But scientists will eventually, health agencies will start testing combinations if it turns out we need it, if we don't get lifelong immunity from this vaccine.
Wouldn't that be nice?
Yeah.
But similar to what you were just saying there, well, what if your first dose is from one manufacturer? Does a second dose have to be from the same manufacturer? So if you got a Pfizer and all there is Moderna left.
I personally would take one or the other because they're so similar. I'd even take a Pfizer followed by AstraZeneca, even though they're different, because in principle, as long as they have the same antigen, you're getting S twice, because the immune system needs to remember the same thing twice. So I don't think there's a problem, but I don't know what the regulatory agencies are saying, because they're the ones who actually make, the guidelines.
Yeah, I mean, it's such a fine balance between, ideally, we wouldn't have to have this discussion. There'd be more than enough vaccine to go around. There wouldn't be variants. There wouldn't be a surge. But I'm glad I'm not the one who has to make these decisions and potentially go against the manufacturer's recommendations. It's easy for the manufacturer to say, you can only do it my way. But at the end of the day, it's such a complex decision, and the calculus involves how many lives are going to be saved if we get this vaccine out sooner than later.
But I do think our regulatory agents have to make those calls, because it's not up to us as individuals to make those calls. The fact is when you put something to humans, it has to be approved by proper clinical trials and approved by the regulatory agencies. And so far, only the same vaccine given twice has been tested. But it is really plausible from our knowledge as immunologists that you could have one dose of one and one of the other and get perfectly good immunity. But I think we should really be looking to our regulatory authorities and our advisory committee on immunization to give us those guidelines.
That makes sense. That makes sense. I like this question here, because it's talking about vaccine hesitancy. So you have a population 20 to 39 who, when they get COVID, it can be a very mild disease. It doesn't mean that they're not at risk for a not very mild disease. Any kind of response back to those type of people that say, well, I'm not really at high risk anyway, so I'm not going to get the vaccine.
Well, I heard a statistic recently that 50% of the people in the ICU were under 65. And there's a considerable number of people who are young who are getting, really severe COVID. And on top of that, we don't know what the long-term effects are of having even mild COVID. So there can be we've heard of this so-called long COVID. I've certainly met people who are in my age group and younger who are now experiencing long COVID. I know of one person. Mm-hmm. So, we just don't know, like, what are the long-term effects of having this virus? And the other thing is that there's some beautiful work by one of our colleagues, Dr. Shane Crotty, who looked really carefully at the immune response in a lot of people of different ages. And what he found was, and his team, was that the variability in the immune response was 200-fold. That's a really big window. And you don't know where you are on that spectrum. And it can depend on how much virus you're exposed to. It could depend on your host genetics, things that maybe you have a mutation you don't even know about. So it's sort of a roll of the dice. So why would you do that when you have a perfectly safe vaccine that will give you immunity? And I know with vaccine hesitancy, there's different ways of approaching that. But I think if we just go to the science, there's a lot of variability in that immune response, and you don't have a guarantee that you're going to be in the right end of that immune response, or that you're not going to have severe COVID or long-term effects from the virus. Mm-hmm. But I get it. It's really, I think we have to approach these people really compassionately as well, because some people are vaccine hesitant for a variety of reasons. So I think probably the first step would be just to ask why, and ask what is it that's keeping them from being vaccinated, and start there.
I would also say that, when you have millions and millions of cases, even a 0.1% death rate in a younger group is a lot of deaths. I happen to personally know, a 35-year-old with myocarditis who months out still can't walk upstairs. So, it's definitely got long-term consequences on people who are, young and healthy. So it's really the weighing the risks of vaccination, which are, very, very low, 1 in a million, versus the risk of severe side effects from an infection that are much, much higher. And I think that's what you have to.
Exactly. And I think the risk of having a low, a few symptoms and you're relatively asymptomatic, and you can then, you're infecting other people. Mm-hmm. So not that a vaccine, stops that, but
it should actually. We hope it will. I just think we need more data.
Yes, cross our fingers. Yes. Wonder if you could talk about using the vaccine in cancer, if someone had cancer or one of those disease states.
Well, the vaccines are completely safe. They're non-replicating, so they're not going to, I think they would be safe in the immunocompromised, whether they have cancer or, they're taking drugs for transplants or autoimmune conditions. We don't know how good their immune response is going to be, but I think they're particularly vulnerable to COVID infection. So on balance, I think it's better to give them the vaccine.
Okay, perfect. Lots of questions. Some questions from me. Actually, someone asked a question about these vaccines. They talk about they're 95% effective. What other vaccines are 95% effective that we already get?
Yeah, I don't know. I think the measles vaccine is at least that good. It's very good. The yellow fever vaccine is one of the best out there. I think that's very high. But then our flu vaccine on a typical year might be 60%. So, it's a variable, and it depends on how infectious the disease is, how many people need to be vaccinated. So for measles, when the vaccination rate drops down below 85, we start to see outbreaks. We don't know what the vaccination rate needs to be for this one. So.
And then finally, I think, can you talk a little bit about the anaphylaxis issue, and what people got severe anaphylaxis from the vaccine, and what was their previous history with anaphylaxis? Was it to foods? Was it to medicine? Was it to other vaccines? One of the questions we ask patients when they get the flu shot is, have you ever had a bad reaction to a vaccine? And when it's yes, I don't know what the other pharmacists do, but I send them off to their physicians
Mhm.
to make that decision, and I don't do that. But what about, like, I was under the impression that it was severe allergic reaction or adverse reaction to a vaccine.
Yeah, the first two Pfizer ones, I think that was the comment. Do you remember, Jen?
Yeah, so the rate of anaphylaxis is still extremely low with these vaccines. It's 1 in 100,000. So I think, like I said, the recommendation is you wait. Well, I think it would be good for them to seek the advice of their doctor, and then you should definitely observe them for 30 minutes. I mean, I assume that that pharmacists have the necessary equipment to deal with
Yes,
anaphylaxis.
they've been trained in first aid and CPR, et cetera, et cetera. So,
but I don't know the answer to what is triggering that. There is this theory that it's the polyethylene glycol is the in part of the formulation. It's not part of the active ingredient of the vaccine, but I think people are probably working on that right now. So I'm sure we'll learn more soon.
Yeah, but I think always referring them to their doctor. Personally, if it was me and I had a history of anaphylaxis, I might want to have my vaccine in a healthcare setting, but you know.
So
yes,
I used to get, I
can,
had an anaphylaxis to eggs, and I used to, whenever I got my flu vaccine, that's exactly what I did. I did it in my doctor's office just to make sure that I didn't, because the flu vaccine, this is not the case for the mRNA vaccines, but the flu vaccines, there's a component, because they're grown in egg, there's always a concern about anaphylaxis. So I got them done in my doctor's office.
Yeah, well, that's maybe the way to do it. Well, we are running out of time, and this has been absolutely fantastic. You guys are great. You've answered so many good questions. I know we'll have more questions, and it'll be more around the actual administration, because we will be presented with so many different types of patients and situations and what they're on, and all kinds of things. I just want the audience to know that we're going to continue this discussion on COVID-19 vaccines and talk a little bit about vaccine hesitancy, because that's a huge issue. You have to approach it the right way. You have to respect the fact that they have there's reasons behind that vaccine hesitancy. Not everybody is going to want to get the vaccine. Everyone should, but then it's a different, it's their decision. And also wanted to let everybody know, again, remind about the pharmacy technician injection training course. So look out for more information coming out from us about that one. We want to get all hands on deck for this one, because there's going to be we have to do 1 million a week or something like that, or maybe more than that, to get a lot of people. So everybody's got to put in, do what they can. Information about when when pharmacists will actually get the shot, that information I don't have. It is on the OPA website, because I know a lot of pharmacists are frontline and they really want to get those shots if they're not in hospitals. Stay tuned for that. That's on the OPA website, as well as how to get engaged in when we might be in pharmacies giving COVID-19 shots. We do know that those COVID-19 shots will be done by appointment based only. So this won't be wander in, hey, do you got any COVID-19 shots could you give me today? So it's going to be maybe a little bit easier, but we just came off a very busy and tiring flu shot. We call it flu season, but it's not the flu season, it's the flu shot giving season. And we had a lot of pharmacists worked really hard, gave millions of doses. So again, I want to thank Dr. Gomerman and Dr. Watts. You've been amazing to work with. You've been brought up all kinds, answered every question that I came up with, and really appreciative. I also want to thank everybody for coming tonight. We had a great turnout tonight. It's been fantastic, and we'll keep on the conversation, because COVID, unfortunately, is still here. So thanks, everybody, and have a great night.
Okay, thank you for having us. Thank you for listening.
You're welcome.